So Dr. Tom Cowan, he's a medical doctor who has co-authored with Sally Fallon Morrell, The Contagion Myth, Why Viruses, including the Coronavirus, Are Not the Cause of Disease. Welcome, Doctor.
Two o'clock. Perfect. Can everybody hear me? I always have to make sure here so I don't fall off the edge. So the first confession I have to make, I was trying to think back in my history. I've done, I don't know how many hundreds of talks. I believe this is the first one I've ever done with slides. So I'm a little nervous about that. Because I think actually all I have to do is push a button, but you never know. I'm actually kind of a Luddite at heart. The second thing I want to say is that for those of you who don't know me, I've been a medical doctor for about 37 years and recently, quote, retired, although I must admit the laws of California Medical Board said that I actually had to surrender my license, which I didn't really like that word. So I tried to negotiate for a different word, but that didn't work. So I actually did surrender my license. And over the years, I've written a number of books. There was 2 in the beginning and then a series of 3. The first one was about why the heart is not a pump, and how blocked arteries are not the main cause of heart attacks or so-called heart disease. So that was the first. And the second one was that vaccines are neither safe nor effective. And the third one was about how cancer has nothing to do with genetics. And so then I decided I was sick of controversial subjects. So I turned my attention to a long-standing interest, which I was trying to avoid for 30 years, which is that viruses have nothing to do with illness and the SARS-CoV-2 virus does not exist. So that was the non-controversial latest book. And so the way this is going to work, we're going to divide this up into 2 half-hour segments. I'll speak for a half hour, then Andy will speak for a half hour, then we'll take a break. And then I'm going to speak for 25 or so minutes, and Andy will speak for 25 minutes, and then we'll have question and answer. And I always like to tell people exactly what you're going to know at the end of this 2 hours. I can't speak for Andy, but I'll tell you what you're going to know from me. I have a little closer. I've been fond lately of saying that if 98% of the people in the room cannot answer this question at the end of this 2 hours, I don't think they should pay me. But that means you have to honestly listen and try to be able to follow the thinking. So the first half hour is very simple. The question which you should ask, everybody in the world should know this, particularly medical doctors and anybody who calls himself a scientist, which is a very simple question of how does a virologist know there is a new virus and prove that it causes disease? Right? If this whole thing is based on a virus and you don't know how to answer that question, which I can literally guarantee, and I hope people check me on this, go to your family doctor and ask them that question and they won't know. And so if you don't know the answer to this question, and you're going to know, A, how it should be done and how it is done, because once you know the answer to that question, you will never believe again that viruses cause any disease. All you have to know, it takes a half an hour, I hope, to answer that question. Then you will know forevermore what the story is, and you will know what your medical doctor doesn't know. Got it? Okay, so here's how you do it. First of all, if you ask most people, how do you know there's a new virus and that it causes disease? They say things like, well, a lot of people got sick in Wuhan, a lot of people got sick in New York City, a lot of people got sick in northern Italy, and so that proves it's a virus. Or they say, well, a lot of people got sick here, and then it spread to the next place, and then they did contact tracing, and so we know that this illness spreads from one place to another, from one person to another, and that's how we know it's a virus. Or they say there was nobody sick at San Quentin, and then somebody had a positive PCR test, and then they sent a bunch of prisoners there, and then they all got sick, and they all had positive PCR tests. That proves it's a virus. Or my Aunt Bessie, God bless her, she was healthy. She went to a church sing, and then somebody was sick there, and she got sick, and she came home, and she gave it to Uncle Fred, and my God, was Uncle Fred sick, and that proves it's a virus. And on and on and on the stories I get. Let's just be very clear. First of all, those are all what are called epidemiological observations. The role of epidemiology in science and medicine is to generate hypotheses, which then can be tested to demonstrate the cause. There is no reputable scientist or medical doctor who knows anything about this who thinks that any epidemiological observation proves the existence of a virus or proves the virus causes disease. Believe me, virologists don't sit around in laboratories watching CNN and saying to themselves, oh my God, a lot of people in Florence got sick. That's how we prove it's a virus, right? And by the way, if you think because a lot of people in the same place got sick, that proves it's a virus, you must think that Hiroshima was a virus. And if you think because the illness spreads from one place to another, that proves it's a virus, you must think that Chernobyl was a virus because it spread from Russia over to Eastern Europe and then Western Europe. And a lot of people got sick, and they got sick in the same way. And that's exactly what people say to show that there was a virus. By the way, for hundreds of years, people noticed that sailors on ships got sick one after another. And then they went to the next port, and then the sailors on the next ship got sick. And for hundreds of years, they thought there was something contagious being passed between one sailor and the next. And then lo and behold, somebody ate a lime or a lemon, and the whole thing went away because it was scurvy. And for hundreds of years, there's a disease called pellagra and beriberi, which killed millions of people and spread in families and in communities, and they thought it was contagious, and there was something transmitted between people, and it turns out it was simple B vitamin deficiencies because they didn't soak their corn properly, and the whole thing went away when they figured out how to eat properly. Which brings up the point that for those who say, oh, Tom, what difference does it make what's causing this? It makes a lot of difference, because if you treat scurvy as a contagious illness and quarantine people, that's a lot worse than giving them a lemon. Or properly soaked corn. So it makes all the difference. This whole thing is based on the existence and the fact that a virus causes disease. And so I will say, I don't need any more people telling me about Aunt Fred or Uncle Bessie, or the other way around, and how they got sick, And that proves, so there, big guy, what do you mean it's not a virus? That is not how we show that there's existence of a virus or that it causes disease. And I would also say, even after 100 years of being in this gig, I don't need any more stories. I agree, we should have tested for some transmissible element. We did, it isn't there, finished. I don't need any more stories. Does everybody have that crystal clear? Nobody's going to say that to me. And you know that if somebody uses that as the reason that it must be a virus, they don't know squat. Got it? Okay. Next step. Now, here is where it gets interesting, because obviously we have to go to a lab. That's what virologists do. Now, here I'm going to make a statement, which I hope, maybe, that you say to yourself when you hear it, are you kidding me? This cannot be true. The statement is, since the electron microscope was discovered, invented in the 30s, which is the only way, right? The only way you can see a virus. There is no other way. There's different techniques of electron microscopy, but that's the only way. And we're going to get into later about how electron microscopy itself is unreliable. But let's forget about that for a minute. Electron microscopy is the way you see a virus. There is not one reported case in 90 years in any medical journal, in any article ever published, of a particle that would be described as a virus being found in any fluid of any sick animal or human being. Did you get that? In other words, you can take, so let's get into some definitions. A virus is a thing, right? It's not, and when I say it's a thing, I mean it's not a feeling or a thought or I have an idea or it's a bad omen or something like that. The conception of it in conventional science and medicine is that it is an actual particle. It has a protein coat and it has a piece of genetic material, either DNA or RNA inside. It is a thing that you can visualize on an electron microscope picture. That thing should be uniform in size and in configuration, i.e. morphology. In other words, it should look the same. It should not be sometimes 2 feet wide and sometimes 6 inches wide. It should not look vastly different. It should be a absolutely uniform, consistent size and shape, and it should have the same genetic material from one virus to the next. Got it? That's what we call a virus. There are maybe hundreds of so-called pathogenic viruses, viruses that cause disease. Not once has anything like this uniform particle been found in any fluid, snot, lung fluid, cerebrospinal fluid, blood, toenail juice, nothing from anybody who's sick. Now, if you don't believe that, let me tell you the people who agree with me on that, right? So I, in a way, would hope, why should I believe this guy? Just because I've been looking at the medical literature for 30 years trying to find such a case. There isn't one. Here are the people who have been asked, can you show me a picture of the SARS-CoV-2 virus? Number one, it's now up to 84 health departments of different countries. The Australian Health Ministry, the CDC, the NIH, Canada, 84 different governmental organizations have been asked under the Freedom of Information request, do you have a single paper showing the isolation of any SARS-CoV-2? That's the name of the virus. COVID is the name of the illness, so-called illness. And they all reply no. Not one case. Here is what in June 2020 the CDC says on page 39 on their COVID manual, quote, no quantified virus isolates of the 2019 nCoV are currently available. As of June 2020, everything that's a thing, like there's 6 chairs, can be counted. Feelings, thoughts, conceptions cannot be counted. Things like viruses can be counted. They have no example of the SARS-CoV-2 virus. The Robert Koch Institute, which is the German equivalent of the CDC, was asked, do you have any paper showing the isolation, the pictures of SARS-CoV-2? Quote, the public relations director, quote, I am not aware of a paper which purified, isolated SARS-CoV-2. Not one paper. Then, in a very interesting twist, the guy whose name is Christian Drosten, he's the Fauci of Germany, he was the one who came up with the original PCR test, right? Here is his paper that described where he got the primer sequences for this new test, for the new virus, right? So now, in order to have a primer sequence of a virus, it's dependent on 2 things. One, that you know that that sequence, that small bit of genetic material, actually came from that virus. In other words, if you find a piece of a hoof in your field and you say that must have come from a unicorn, it would be nice if you actually had analyzed a unicorn, right? If you've never seen a unicorn, it would be very difficult to make a compelling case that you know that was from a unicorn. And then you have to show that no other animal has that same piece, right? Otherwise it wouldn't be a unique test for a unicorn. So here's what he said, quote, the title, Detection of the 2019 Novel Coronavirus by Real-Time RT-PCR. This was the start of all the PCR tests done in the world based on these primer sequences that were coming from the SARS-CoV-2, the virus, unique to that virus. Here's what they said, quote, we aim to develop and deploy robust diagnostic methodology for use in public health laboratory settings without having virus material available. They never saw or had the virus. They only saw it in the computer. We'll get into that a little bit later, how they make that. They never had an example of the virus. Who else agrees with this? Well, the virologist who wrote the 6 leading papers describing the isolation and finding of the novel SARS-CoV-2 in this corona crisis, all admitted in writing when asked, did you isolate the SARS-CoV-2 virus from any sick person, said unequivocally no. We never isolated, we never found SARS-CoV-2 from any sick person. And then Andy and I had the privilege of presenting to a group of very prominent anti-vax doctors, lawyers, activists, etc. And They were not happy with what we were saying, and so they invited 2 virologists and a third guy who was introduced to us as 20 years senior laboratory virologist at the Wuhan's Chinese Center for Disease Control, and then 20 years senior pathologist doing virological laboratory work at Yale University. And he was there to debunk us. Tom and Andy are nuts. And I thought, this will be interesting. And he agreed that there is no case of SARS-CoV-2 being isolated or found from any sick person, period. What's the reason for this? Why can't you find it? Because, I just want to be clear on this, it is not that things the size, shape, and complexity of viruses cannot be found. We've been finding things called bacteriophages since the 30s. You take a culture of bacteria, you macerate it like in a blender, you filter it, you centrifuge it, it comes out in a band. You see these particles, the size, shape, morphology, and complexity of viruses. They're all the same geometry, same morphology, same consistency. You can do that with bacteriophages, you can do it with giant viruses. It is not a technical problem to find a virus in a bodily fluid. That is not the problem. So we asked him, why can't you find it? And he said, there's not enough to find. And luckily, Andy asked him, so if you had 10 people and you mixed their sputum together, would there be enough to find then? And he said, no, not enough to find. How about 100 people? No, not enough to find. By the way, if there's not enough virus to find in 100 people, what is the theory that it's going to kill us all? So then we kept going, 1,000 people, no, not enough to find. Then 10,000 people, not enough to find, no more questions. So we don't know if 100,000 people there would be enough to find. So if you think that virologists find the virus in any fluid of any sick person, you are misinformed. That's not how they do it. Nobody disagrees with that. Nobody. They will tell you they do. They will tell you, oh yes, it's in the, of course we can find it. Of course. Somewhere. But no, you can't find it. There's 2 reasons why they say. One, there's not enough to find, which of course, then how is it going to kill us all? The other reason that people have said to me is, well, that's because viruses are intracellular parasites. They only live inside the cell, so obviously you can't find it outside the cell. So I asked them, so how does it get from one person to another? Well, it gets outside the cell and it goes to the next person. So why don't you catch it then? Well, it's not there for very long. I said, look, hire Fred, this guy over here, tell him every 15 minutes, go look, if this guy's sputum. It'll come out 15 minutes, you catch it then. Nope, Fred's too expensive. Anyways, he sleeps at night. So that is how you should find a new virus, right? You take a sick person, you do standard virology techniques that have been around since the 30s, macerate, filter, ultracentrifuge, comes out in a band, suck the band out, you show that it's a uniform morphology, has the same genetic material, you can sequence it, you can get sequences from it, you could even make primers from it if you had it. You cannot make primers if you don't have it first, right? You cannot say this piece is from a unicorn if you've never seen a unicorn. That's what we call common sense / thinking. So the question is, why are there 10,000 approximately papers with the title, The Isolation and Purification of such and such a virus, including, let me guess, 50 SARS-CoV-2? There are such, and people who will want to dispute you will say, here's a paper, New England Journal, here's a paper, Nature Magazine, Isolation, Purification of SARS-CoV-2. Never mind that those people said that they never isolated it from a sick person. But obviously the paper is there. How did they do this? How did they do this? Why are they saying this? So this technique that 100% of virologists use, 100% of the papers with that title do exactly what I'm going to say now. They take the fluid from a sick person, usually one person, with supposed alleged new disease like COVID. They take the lung fluid, they macerate it a little bit, they sometimes centrifuge it or filter it, and nobody says that's purification. Now you have anything liquid from somebody's lung, right? Nobody says that's the virus. And this got started by a guy named John Franklin Enders in 1954, wrote 2 papers on it that started or rescued virology. Virology had gone 20 years proving you could never find the virus in a sick person. And he saved the day. Here's what he did. He took fluid from somebody with measles. Now we take fluid, the lung fluid from somebody with COVID. We macerate it, we filter it or centrifuge it. And then, according to Enders in his paper, he mixed it with bovine amniotic fluid, beef embryo extract, horse serum, antibiotics, phenol red, soybean trypsin, milk, and that was it. Now, all of the game we're playing is find the piece of particle with genetic material. We now have 6 other things with particles and genetic material. And then he takes that mixture and inoculates it, means spreads it on monkey kidney cells. And then he puts in 2 antibiotics. Now we use amphotericin, otherwise known as amphotericin and gentamicin, and we mix those in, and then the kidney tissue breaks down. It's called the cytopathic effect, CPE. And then it breaks down into little particles. And that is the proof that the virus killed the kidney tissue, the virus exists and is the pathogen. Did you get that? That is not a proof. That is the proof that there was a virus in the measles person called the measles virus. And that measles virus killed the kidney tissue, right? That is the proof. It's called CPE, cytopathic effect. And then they show you the pictures of the particles that were generated from the killing. Everybody got that? That is called the isolation of the virus. Now, interestingly, in 1954, in his original paper, Enders said something very interesting. 2:30. A second agent, I'm quoting now, a second agent was obtained from an uninoculated culture of monkey kidney cells. The cytopathic changes it induced could not be distinguished from the viruses isolated from measles. Let me say that in English. He decided to do the same experiment, I think, because he didn't actually write down what he did, without putting anything from anybody with measles. He just added the bovine serum and the horse serum and the antibiotics, and he inoculated that onto the monkey kidney cells. And you know what happened? The same damn thing. And he said, you guys better check this someday, because I think maybe this was coming from the way we did the experiment. And for 63 years, nobody did. Until last month. And I'm not going to show you that slide, but a friend of ours, Stefan Lenka, with our support, I would say, decided to run this experiment again and see if you just add nothing, whether you can get SARS-CoV-2 from just monkey kidney cells, gentamicin, amphotericin, and fetal calf serum, and it turns out you get the same cytopathic effect, which is the proof that viruses exist and cause disease. And it's 2:30. If you could put that slide up, I just want to say one more thing. So this is the slide. Andy's going to go over this. So let's go to the next one. So this was, I'll tell you in a minute. Can you hear it? Read this. Can we stop it? So Montagnier was asked, did you ever find, he was the guy who got the Nobel Prize for discovering HIV, right? And what happens when you kill the cells is you get cellular debris, all these different size particles. And he says, the guy who did his electron microscopy, we never found a virus. All we found was cellular debris. And that's what Montagnier said. And then, just to finish, you will see, people will show you pictures of the virus, right? You've all seen it. And that comes from the breakdown of the kidney cells in the culture. Never, never, never from a sick person. Now, I don't mean this thing. This was made by the graphics art department of Sussex University sometime in 2019 to scare the shit out of everybody. I don't mean this one. This is another picture that they made. I mean, because if you believe those are proof of the virus, then you obviously believe that's proof, or that is proof that unicorns. I mean this guy. You've all seen that? Proof. And that tells you that you have identified the virus. There's another one. What about all these other things over there? So this paper, I won't go into it, says, this was by kidney specialists saying, we see these exact same particles from back from 1990s in kidney biopsies. They have nothing to do with a SARS-CoV-2. These are viral-like inclusions that were found in all 15 cases. They are mimickers of SARS-CoV-2, and, the CDC knew about them in 2003, and we even know what this so-called spike protein, those little dots, turn out to be a protein called clathrin, and they are not have anything to do with any new virus. The final thing, this was a CDC paper. It turns out that when you grow this virus, so-called grow the virus on different cells, you get radically different composition of the virus. How can that be? If you grow chickens with one food and then you put them in different food, you don't get horses in one and chickens in the other. The only explanation for why the protein composition of SARS-CoV-2 is completely different depending on what you grow it in. This is cellular debris. It's just garbage coming from the breakdown of the cells. And I will finish with one thing. This is exactly like an analogy that you may have heard. It's like you go around your neighborhood and you see little bits of paper and you think the paper, little bits of paper, and then you come back in a week and the lawn is filled with little bits of paper. And you say, I know what happened. The paper inoculated it inside to the house. It took over the machinery of the house, had its way with the house, reproduced itself, budded out from the house, blew up the house, scattered paper all over the lawn, and that's why we find it here. And some little boy on a bicycle comes by and says, Tom, somebody came by with dynamite and blew up this house. That's what happened. We blew up the house by starving it and poisoning it, and all that is just debris coming out of the cells, and from now on, you will not believe this nonsense ever again. Thank you, and